request one of you to give us a prayer. One of you to pray for us. Yes, Nasuna.
Let's turn ourselves forward of prayer. O Lord Jesus Christ, one, thank you for this day, one, thank you for the gift of parents and education plus the teachers. O Lord Jesus Christ, pray that you guide us through this lesson. Pray that you may understand everything we are going to learn, and we pray that you get the courage to ask what you are not understanding. Jesus' name I pray, amen.
Amen. Thank you so much for the prayer. And it's my prayer that you shall allow for good network. I know my colleagues will be in, and I'm the lead teacher today. We are continuing from where we stopped last time. Remember, we were taken through transporting plants, and more specifically, we're looking at some aspects of transport. Those are terms that are involved. The teacher emphasized on the definition of osmosis, having done diffusion earlier. And then the focus here was made on the movement of solvent molecules. Another term that we were saying is not missed in the definition is a selectively permeable membrane. And by definition, a selectively permeable membrane is that which can only allow smaller molecules to pass through. Large-sized molecules cannot pass through a selectively permeable membrane or semi-permeable membrane. So that is one of the terms that should not miss in your definition when the teacher was emphasizing. And he emphasized the point that it is the movement of solvent molecules. And then the last part that was emphasized was that this movement should be from a dilute solution to a more concentrated solution. You could also look at it the other way around. A dilute solution is that which has a higher concentration of water molecules. And then a more concentrated solution is that which has lower concentration of water molecules. So if you are to define it in terms of the concentration of water molecules, then you could look at it in terms of movement of solvent molecules or water molecules from that area where the water molecules are of higher concentration to that area where water molecules are of lower concentration. It is the same, only that here you could be looking at it in terms of the water molecules. And then he went ahead and you discussed about movement using artificial membrane, movement of water molecules or solvent molecules using artificial membrane. And here an example was a Viskin tubing. And we observed that when a tubular structure, a tube is made out of the Viskin tubing, and you place a solution in that tube, eventually we see water molecules moving into that tube. And the effect is that the tube is going to swell, is going to increase in size. So again, that movement of water molecules from outside into the tube or from a dilute solution or pure water into the Viskin tube that contains the salt solution or sugar solution is by the process of osmosis. Then I remember you had discussed about the movement of water molecules in a living tissue. That is a movement demonstrating osmosis using a living tissue like an iris potato. So Irish potato, and then a cup kind of shape structure was made out of it. And you observed that water could move from outside into that space that contains salts. So I want to pick from there and share with you a simple finding that I observed last Sunday when I was going to the church for prayers. I stay in a place near a primary school where I just crossed from where I'm staying to the parish. So I observed some two kids who were excitedly looking at a snail, and one of the kids, I stood near them, and one of the kids ran into their home and brought some salt. According to him, his teacher told him a story from school, and he wanted to investigate it. So he put that salt onto the snail that was moving, and soon they observed something funny. Probably this is a similar story that you could have seen somewhere. Maybe one of you could share with us that if you had ever seen it, and then we pick from there. Anyone who had seen a similar experience with a snail coming into contact with the salt? Yes, I see Paula. Would you like to share with us?
The snail slowly dissolves.
Sorry?
The snail slowly dissolves.
The snail slowly dissolves. As it is dissolving, what do you see outside? Outside the snail's body and on the salt, what do you see?
Like juices that the
Water.
You see water. Am I hearing from Melda Nakaita? What do you eventually see outside the snail's body and on the salt?
Water molecules.
You see water molecules. Where could this water be coming from?
From the snail.
From the snail. And what happens to the salt outside?
Dissolves in water.
It dissolves in water. Very good. So that is the experience that I want us to pick from. So the snail, in a nutshell, is made up of tissues. It's made up of cells. So when you see water moving out of the snail into the salt, then the salt dissolving means there is movement of water molecules from the tip of the snail to outside where the salt is. And when the salt dissolves, actually it forms a very concentrated solution outside. So for me, that is the same process of osmosis that we are looking at. So it goes to answer the question, does osmosis occur in animal tissues? And the answer is yes. We can take that deep into the tissues, into the cells. If we were at school, we would have demonstrated it by getting red blood cells from our rats. Then you would place them into solutions in a beaker or solutions in a test tube. Then you will observe what then happens. So allow me to take you to what happens with the cells. Animals in inferior solutions.
What is this I'm seeing on the screen? How do they screenshot?
What is wrong with you, teacher?
Mama, what is wrong with him?
How do they swim?
I tell you nothing his son. You say you can run away. Mama. You see the son, trust me, you run away.
Trinity, mute. You're unmuted.
Finish.
Mama.
Sorry for that glitch. I was trying to connect to this. Osmosis using red blood cells. And I was saying, if we were at school, we would have demonstrated this using red blood cells from the rats. Then we would have placed the cells into various solutions, then see what happens to it. May I request one of you to read for me that? Mute yourself and read for me.
Placed in a solution with a lower solute concentration than is found in the cells, water moves into the cells by osmosis, causing the cells to swell and eventually burst. Hemolysis. A hypotonic solution is one which is dilute compared to another solution.
Yes, thank you. When you place an animal cell into a solution that has a lower concentration or in pure water, just like what happened in the snail cells, then we expect this cell to absorb water. And as it absorbs water by osmosis, then it is going to increase in size until something else happens. So another person to read the next.
Chami.
Go ahead.
When the red blood cells are placed in more concentrated solution than is found in the cells, hypotonic solution, water moves out of the cells to the surrounding solution by osmosis, causing the cells to decrease. They shrink or appear wrinkled. This is known as crenation.
Yes. So there are two things that are happening here, depending on the nature of the surrounding solution. Let's go back to the first one. What eventually would happen to these red blood cells that are placed in pure water or in dilute solution as they swell? What do you think will eventually happen to them?
They will burst.
They'll burst.
Very good. They will burst. Why do you think they will be able to burst eventually? What will make that possible?
The water will move into the cells by osmosis.
True. True. Meaning that the tissue that is forming the cytoplasm, rather the plasma membrane of the red blood cell, can only extend up to a certain point, beyond which it can no longer sustain stretching. So because of that, it will eventually burst. And the term used here is it gets hemolyzed. Then for the second circumstance, it is where the cells are going to lose water, and then they will reduce in size. and then we say eventually they are created. Then there is the third scenario. The first one is where you're placing it into a concentrated solution. Then the third one is where the solution inside and outside of the same nature. So allow someone to read for me the last part.
However,
Go ahead.
when red blood cells are placed in isotonic solution, they neither gain nor lose water. Isotonic solution is one with the same solid concentration as a cell cytoplasm.
Thank you. So please take note of those three types of solutions. One is a hypotonic solution. By definition, a hypotonic solution is that whose concentration is lower than that of the tissue placed in it, or lower than that of the cell placed in it. I repeat, by definition, hypotonic solution is that solution whose concentration is lower than that of the tissue or of the cell that is being placed in it. So hypotonic solution always loses water to the tissue that is placed in it. And then second one is hypertonic solution. By definition, is that solution whose concentration is higher than that of the tissue or of the cell that is placed in it. So hypertonic solutions always gain water from the tissues that are placed in it. So it means the tissue that is placed in it is going to reduce in size. Then isotonic solution, similar concentration, like all the tissues or the cells that are placed in it. Therefore, a tissue that is placed in an isotonic solution has no net movement of water molecules, meaning that the number of water molecules moving from the surrounding solution into the tissue is the same number that will move from the tissue into it of the water molecules. So the three types of solutions are demonstrated here, and what happens with the tissue placed in it. So the first one is hypertonic, where it gains water from the tissue or from the cell, and then the consequence is that the cell reduces in size, so the cell gets created. And then the second one is isotonic, having similar concentrations, and therefore there's no net movement of water molecules either into the tissue, into the cell, or outside the cell. And then the last one is hypotonic, where the cell gains water, and because it can no longer continue to expand, eventually it bursts. So, given the this is still demonstrating what happens in those beakers. And then this one is showing the movement of the water molecules. So in a similar way, you could also look at the movement of water molecules in relation to plant cells. Somebody to read for me that? Unmute yourself, please.
Plant cells are rigid
Plant cells
cells.
possess a rigid cell
One
wall
person,
which
please.
Plant cells possess a rigid cell wall which prevents the cell from bursting. When a plant cell is placed in a dilute solution or water, then the cell sap, water enters by osmosis through the cell membrane into the cell sap. The volume
Yes.
of cell sap increases and it makes the sap vacuole expand. This causes the protoplasm move towards the cell wall, thus gaining turgidity.
Yes.
Time comes
Time comes when the protoplasm is pressing against the cell wall and no more water can be absorbed. At this state, the cell is said to have gained full turgidity, and the force on the cell wall is called turgor pressure.
It's called the turgor pressure. Yes, unlike the scenario we saw with the animal cell, when you are looking at the different types of cells from senior 1, you realize that plant cells have outer part called the cell wall, and that cell wall can be so rigid, meaning that it cannot expand, especially in an old plant cell. It cannot expand, therefore it will have restrictions on the expansion of the cytoplasm from inside. So, which is not the case with the animal cell, which is lacking the rigid cell wall. So that explains why for the animal cell, it can expand, eventually it will burst. While for the plant cell, it will expand, and when the expansion reaches the cell wall, then it is going to experience some resistance from the cell wall, which will prevent further expansion. So at that stage, we say the cell has become turgid. And that's the same concept of movement. The movement of water molecule from outside the cell into the cell is by the same process of osmosis. And that is what happens when you plant your seedlings into the soil. They are going to absorb water. When they absorb water, that water goes right into their cells. And for the young plants or the seedlings that you have got, they are going to rely on the turgor pressure to gain some kind of support. So in the application of this, we see that turgor pressure is important, especially in those plants where the supportive structures are still developing. And it is based on the fact that you're going to see the cells expand until when they experience some inward resistance by the cell wall, what we call the turgor pressure, and it is this that will be used as the way of support in the plant. Somebody to read for me the last one.
Plant cells possess a rigid. Therefore, the last paragraph.
That's when the cell is placed. Somebody, please. When the cell is placed in a more concentrated solution, yes.
When the cell is placed in a more concentrated solution.
One person, please.
When the cell is placed in a more concentrated solution than its cell sap, water moves from the cell sap through the cytoplasm, then the cell wall to the surrounding solution. This or plasmolized. Therefore, a flaccid cell is one whose protoplasm has lost contact with the cell wall due to loss of water from the cell sap of the vacuole by osmosis.
Okay, so there are terms that have been highlighted here. So when you place a plant cell in a more concentrated solution, just like the animal cell, that plant cell is going to lose water by osmosis to the surrounding solution. And when it loses water, then the cytoplasm is going to shrink. And as it continues to shrink, the cell membrane is going to pull away from the cell wall. Then we say the plant cell is becoming plasmolized. And as the plant cell is becoming plasmolized, then it loses its turgor pressure. So plasmolysis simply means the plant cell losing water to the surrounding solution by osmosis, and in effect, that plant cell becomes flaccid. I'm getting feedback from somebody's mic. Kindly mute when you're not reading. Mute your mic, please, if you're not reading. Hello? Okay. So this is an illustration of the three circumstances. The first one is when we place it in a plant cell in what we call hypertonic solution. We said it will lose water. And then the cytoplasm pulls away from the cell membrane, pulls away from the cell wall, and that begins the process of plasmolysis. And then depending on the concentration of the surrounding solution, the cell membrane may pull completely away from the cell wall, cell wall. Then for the case of the isotonic, again, I'm using the term, the reference, the phrase net movement. In the isotonic solution, there is no net movement of water molecules. In a hypertonic solution, there is net movement of water molecules from the plant cell to the surrounding solution. And then for hypotonic solution, the last one, there is net movement of water molecules from outside into the plant cell. When we talk about net more movement of water molecules from one side to the other. So for the case of isotonic, it means the number of water molecules moving into the plant cell and the number of water molecules moving from the plant cell to the surrounding solution, they are the same. For the case of more water molecules moving from the plant cell to the surrounding solution, that is the net movement. And then for the case of hypotonic, it means there are more water molecules moving from the surrounding solution into the plant cell. So that's why we talk about net movement. So net movement means movement above, more. I hope we are able to get that. This one is illustrating the nature of the plant cells, a plasmolized plant cell and a turgid plant cell. So plasmolized, that which has lost water, turgid, that which has gained more water. So it becomes turgid, and that is the basis of strength in non-woody plants. How about the plasmolized cell? When does this occur in nature? This occurs especially in plants that lose a lot of water to their surrounding. And that is especially in those plants that are wilting. In dry seasons, plants wilt, meaning that they lose more water to the surrounding than the water the water that they gain from the soil. So because of that, the experience, the circumstance that happens in plasmolysis. So when plants wilt, it means they are losing more water to the surrounding than they gain from the soil. So because of that, they normally drop The word we use is drop. So this is what happens in a plasmolized cell. We can see the protoplasm pulling away from the cell wall.
Excuse me, teacher.
Yes, I think I can pick questions here.
Teacher, I'm confused. I don't know. I did hear you when you were talking about the definitions of hypertonic and
okay. There are three types of solutions that we could have, and we are looking at them in relation to the tissues. So if you are talking about the tissues or cells, and then you could be looking at a part of our bodies, for example, part of plants. So where we are here, we are trying to pick a part of a plant, and then we'll put it in a given solution, or part of an animal, a tissue, and then we'll place it into a solution. We want to see what happens when we place a tissue like blood into those three types of solution, or what happens when you place a tissue from plants. Maybe you cut part of a fruit, you cut part of a root, you cut part of the stem, and you place it in any one of those three types of solutions. So we have identified the three types, and we are comparing the concentration of these solutions to that of the solution that is found inside the tissue. If you're looking at the plant, then you're looking at the concentration of the cell sap in the plant cell. If you're looking at an animal tissue or cell, you could be looking at the concentration of the solutes within the cytoplasm. So we said a hypotonic solution is that solution whose concentration is lower than that of the tissue that you are placing in it. If you place an animal cell in it, then you're comparing the concentration of the external solution with the concentration of the solutes inside the animal cell. If you have placed a plant tissue, then you're comparing the concentration outside the plant tissue with that inside the plant tissue. So hypotonic solution is that which has a lower concentration as compared to that concentration inside the tissue that you've placed in it. And then the reverse is true for hypertonic. Hyper is higher, hypo is lower. Hypertonic solution is that whose concentration is higher than that found inside the tissue which is placed in that solution. If you're looking at a plant tissue, it means that plant tissue must be having a cell sap whose concentration is lower. Hypertonic, higher concentration. Hypotonic, lower concentration. So hypertonic solutions tend to extract water from the tissues. Hypotonic solutions tend to give up water to the tissues. Then the third one is isotonic. Iso means the same. So meaning that isotonic solution is that whose concentration is similar to that of the tissue which is placed in it. So if you're looking at it in terms of movement of water molecules, it means the number of water molecules moving from isotonic solution to the tissue is the same number of water molecules moving out of the tissue into the surrounding. So those are the three types of the solution. So maybe we can now bring in the demonstration here. An experiment to demonstrate Taga pressure probably, and the plasmolysis in a plant tissue. May I request one of the guys to read for us the materials and the procedure?
Materials: cork borer, test tubes, water, Irish potato, razor blade, and sucrose.
Thank you.
Procedure.
Yes, go ahead.
Procedure. Step 1. Get 4 test tubes and pour 3/4 of water in 3 of them and leave 1 empty. Step 2. Mix the sucrose in 1 test tube to make 5% sucrose solution. Step 3. Mix sucrose in another test tube to make 50% sucrose solution. Step 4. Leave 1 test tube with pure water and the 4th test tube empty. Step 5. Using a cork borer, make 4 potato cylinders of the same length, for example, 3.0 centimeters. That's 3.0 centimeters. Name this the initial length.
Thank you so much. Before we go to next slide, there is something that I want you to take note of here. Later on, relating to question how we can answer questions here, you could be asked to describe an experiment to demonstrate maybe Taga and plasmolysis in a plant tissue. And the approach to it will be the way it is outlined here. So we are going to begin with the aim of the experiment. That is an experiment to demonstrate Taga and plasmolysis in a plant tissue. Then from that, you go to the materials that you will need. Then number 3, you go to the procedure. And then number 4 is what is appearing on the next slide. Continue reading that statement.
The potato cylinders in each test tube or beaker and leave the setup to stand for about 30 to 40 minutes.
Yes, that is still part of the procedure. Then this will be followed by your observations and then conclusions. Allow me to take you back. When you are making solutions using 5% sucrose, 50% sucrose, and then pure water, what do you say with in terms of the concentration? How does 5% sucrose compare with the 50%? Which one seems to be more concentrated?
50%.
50%.
More concentrated
Thank you. Thank you. Then we could also deceive you by saying, by giving you another concentration, which is 0% sucrose. What does that mean in terms of sucrose dissolved in water?
It means that there is no sucrose present.
It's only water.
Isotonic, no sucrose in water. Can we have one person? 0% sucrose solution. What does it mean?
Yeah, it means there is no sucrose present in that test tube, I think.
Very good. It means there's, yes, it means there is no sucrose in it. In other words, you're talking about pure water. Pure water. Okay, so take note of that. So here we have the beakers with the solutions, and then outside the tissues that have been cut after using the cork borer. And then they're supposed to be placed in the test tubes that are on the right here. And then after leaving the setup for 30 to 40 minutes, we could have our results. So remove the cylinders from each of the test tube and measure each length. Also fill the texture, and then tabulate your results as shown in the table below. So those are anticipated in pure water. we see final length 3.3. In 5% sucrose, we see the final length 3. In 50% sucrose, we see the final length 2.8. What do you see happening as you move from pure water to 50% sucrose solution? What do you see with the final length? Is it increasing? Is it the same? Is it decreasing?
It's decreasing.
Do we all see that?
Yes.
When you move, when you are measuring the length of the tissue, beginning with that in pure water to a solution, sucrose solution of 50, we see the concentration of the external solution is increasing, while the length of the cylinders, the tissues, the length is decreasing. What does that therefore mean in terms of water in the tissues? Are they gaining water? Are they losing water?
Gaining.
Think about it again. As you move from pure water to 50% sucrose solution, do you think that these tissues are gaining water or they're losing water?
They are losing water.
Losing
water. Very good. They are losing water. It means they are reducing in size. As they reduce in size, that is because of the loss of water to the surrounding solution. So meaning that 50% sucrose solution can comfortably be described as a hypertonic solution. Which of these three solutions could be an isotonic solution? When you compare the initial length with the final length, pure water, 5% sucrose solution,
5%.
then 50% sucrose solution. Which one could be isotonic? Sorry?
Empty beaker.
5% sugar solution.
Empty beaker.
5% sugar solution.
Girls, let's move together. I've not gone to the empty beaker. I've stopped at 3. So pure water, 5% sucrose solution, 50% sugar solution. You're comparing the initial length of the tissue with the final length. Which of these three could be an isotonic solution?
5% sugar solution.
5% sugar water.
Is it?
5% sugar solution.
5% sucrose solution. Why do you suggest so?
Because there is no change in length.
Because it didn't change in length.
Very good. There is no change in the length of the tissue. That means there was as much of water molecules that moved into it as the number of water molecules that moved out. So in the end, the length remained the same. And then we have already talked about 50%. We said that is a hypertonic solution. Therefore, pure water must be a hypotonic. If we didn't know that it was pure water, and then we could say it is a pure solution. Since we know it is a pure water compared to the tissue, we say therefore it is a hypotonic compared to the tissue. Now let's look at the tissue in the empty beaker. What happened to it?
It reduced. It decreased.
It decreased. You remember the tissue was placed in a test tube without water. What do you think happened?
It lost water to that atmosphere.
Very good. It lost water to the atmosphere. Which process could that have been? Loss of water to the surrounding?
Evaporation.
Very good. That is the process of evaporation. Does this happen in our homes, at home? For example, what happens when you bring vegetables and you place them on the table and you leave them for some time? What do you normally see happening to it?
They dry up.
They dry up.
That is
They don't look
precisely.
fresh.
They don't look fresh. They dry up. That is precisely what is happening here. So when you cut the tissue and you leave it exposed, it is going to lose water to the surrounding by evaporation. Therefore, in the end, the length of the tissue is going to reduce. Now, based on those findings, we can now calculate the change in length by subtracting. That's how we got those figures under the change in length. So in pure water, we have got 0.3. In 5% sucrose, we have got 0. In 50%, we have negative 0.2. And then in empty beaker, we have negative 0.1. And the percentage change is obtained by dividing the change in the length by dividing it by the initial length, then multiplying it by 100%. That gives us the percentage change in the length. So for pure water, it is 10%. Then for 5% sucrose, it is 0. 50%, it is a negative 6.67. Then for empty beaker, the one in empty beaker, it is a negative 3.33. The negatives tell you that it is loss. Then the positive ones indicate there is gain or any increase in the length of the tissue. Negatives mean there is a decrease in the length of the tissue. So on the other hand, when you feel feel using your fingers, those tissues from the different solutions, in pure water, it will feel hard and rough. In 5% solution, it will feel rough and hard. Then in a hypertonic solution, that's 50%, it will feel soft and smooth. While in an empty beaker, it will also feel soft and smooth. In other words, a tissue that has lost water will appear flabby, and the cells will appear flaccid. There are those two terms I'm using. The tissue appears flabby, feels smooth, and soft. When it gains water, it is hard and rough. So remember those two descriptions: rough and hard, soft and smooth. Rough and hard for where it has gained water, soft and smooth where it has lost water. So observations for this experiment. Allow one of you to read for me through those observations. Go ahead.
The cylinder in water had increased in length and become tougher. The cylinder, isn't it? The potato cylinder in water had increased in length and become tougher. The potato cylinder in 5% sucrose solution didn't change in length and the texture remained the same. The potato cylinder in 50% sucrose solution had decreased in length and become soft, flaccid, and curved. The potato in the empty beaker decreased in length.
Okay. Conclusion? Finish with it.
Conclusion: turgidity and plasmolysis occurs in plant cells.
Thank you so much. So that marks the end to that particular question that I was talking about, where we're supposed to describe, please remember the title or the purpose. You remember the materials that you need. You remember the procedure. You remember the observations. And finally, you should be able to remember the conclusion. That's how you should be responding to it. Then the explanation for our observations, another person to read for us.
The cylinder in water increased in length because water in from the surrounding water by osmosis, because the cell sap had a higher concentration than the surrounding water. There was length for the cylinder in 5% solution because the solution had the same concentration as the cell sap of a potato cylinder, hence net osmosis. There was a decrease in length for the cylinder in 50% sucrose solution because water molecules moved out of the cylinder, which had a lower concentration by osmosis.
Thank you so much. So that is an explanation of our observations. The last part, you can read that, the last part.
There was a decrease in length for the cylinder in the empty beaker by evaporation.
Okay, so there was loss of water by evaporation. The last part of it is the significance or importance of osmosis in plants. I'll ask one of you to read them for us. Those are straightforward.
Absorption of water.
Yes, absorption of water.
Significance of osmosis.
One person, please.
Absorption of water by
Significance of osmosis in plants.
Yes. Number 2.
Absorption of water by
It enhances movement of water from root hairs via the cortex to the xylem.
Allow me to pause here a bit. So later on, when we'll be looking at the movement of water through the plant, you actually say that it is because of the
significance of osmosis in plant.
Hold on, please. Movement of water through the plants from the
Absorption
soil.
of water.
Hold on, please. Second point I want to.
From soil via the cortex to the xylem. It
Okay.
facilitates.
I was trying to shed more light on the second point, please. Allow me to do that.
Okay.
So I was saying the movement of water molecules from the root hairs through the plant into the atmosphere eventually involves the process of osmosis. And it therefore means there must be a concentration difference between the salts in the root hair compared to the soil, so that it enhances uptake of water. Then there is movement of water from the root hairs across the cortex into the xylem, which again also involves the process of osmosis. So you remember that it is important in that movement. The third point, please.
Or support in non-woody plants.
Yes, we talked about the turgidity. The last one.
It facilitates opening and closing of stomata.
Yes. Opening and the closing of stomata is because of the behavior of the gut cells. So the gut cells have the vacuole like any other cell, and they may absorb water or release water depending on the sugars in it which are made during the day. Then osmosis in the other organisms. Another person to read for us those 3 points.
Significance of osmosis in other organisms.
Yes.
Movement of water into unicellular organisms.
Yes.
Movement of water from tissue fluid to the cell. It enables reabsorption of water into the bloodstream via the kidney tubule.
So all those involve the process of osmosis. The last one that I want to talk about is active transport, unless there's a question here. Mr. Bagambisa?
Mr. Bima? Mr. Bima?
Bagambisa, are you still there?
Mr. Bima?
Yes. I cannot see hands here. If there are any hands raised up, you let me know. Any question so far? Okay. So I'll introduce this one, active transport. Remember the concepts that have been talked about so far. You have diffusion, you have osmosis. The last one that we're talking about is active transport. One person to read for me that definition for active transport.
Active transport is a movement of dissolved molecules into or out of a cell through the cell membrane from a region of lower concentration to a region of higher concentration. The particles move against the concentration gradient using energy released during respiration.
Okay, so important in the definition, movement of substances, movement of molecules, movement of dissolved molecules, concentration to a region of higher concentration through a semi-permeable membrane. Most importantly, it involves the use of energy. So use of energy, the direction of movement, and the concentration difference should be brought out in your definition. So importance of active transport and
importance of active
Importance of active transport. Used by plant root hairs to absorb mineral ions from the soil. Is mineral ions salt? Importance of active transport. Used by plant root hairs to absorb mineral ions from the soil.
Hey, can we have one question at a time?
Importance of active transport. Used by plant root hairs to absorb mineral ions from the soil. These mineral ions or salts are responsible for setting up an osmotic potential in the root hair, sap, responsible for osmotic water absorption by the.
Yes, Mr. Bima, the time is past spent, so we need to find, we need to wrap up.
Yes. Somebody to read that for me, please. Importance of active transport.
Used by plant root hairs to absorb mineral ions from the soil. These mineral ions, salts, are responsible for setting up an osmotic potential in the root hair sap, responsible for osmotic water absorption by the plant.
Thank you. So I want to re-emphasize this. For active transport, it involves movement from a less concentrated to a more concentrated. And that is what happens when plants are absorbing mineral salts from the soil. For the absorption of water from the soil, remember it is basically because of the process of osmosis. But for uptake of mineral salts from the soil, it is basically because of active transport, and that involves use of energy. Therefore, anything that affects energy is going to affect the process of active transport. Anything that affects the process of energy production or tissue respiration, therefore, is going to affect the process of active transport. And in relation to the plants, it's also going to affect the uptake of mineral salts, and eventually the plants that live in those areas are going to be affected. Somebody to finish reading those three.
Importance of active transport.
Yes.
Contents of active transport used in the absorption of food materials from the ileum into the bloodstream, used in the reabsorption of minerals in the kidney during urine formation, used in the secretion and active uptake of ions in the fish gills from fresh water.
So those give you some areas where active transport is involved. In the ileum, where it involves absorption of food substances, in the kidneys, and then in the process of secretion by the cells. I think this is where we're going to end. So what happens with the plants that grow in areas full of water? Right now, we're talking about land that is being swallowed up by water, flooded areas. Flooded areas, eventually you realize the plants, as much as they are in water, they seem to be drying up. So the question is, what happens to those plants? Any idea from you that you would like to suggest? Those plants, they are still in the water, but they seem to be drying up. What is happening?
Teacher, can I check?
Yes, go ahead.
The plants absorb the water, and maybe the water fills up the that other things should have filled.
The Abima, the time is past spent.
Yes, please. We're finishing with this particular part. Yes. Would you like to explain more?
Teacher, that's where I end.
Okay.
I think.
Yes, go ahead.
I think the cell sap in the plant is less concentrated than the mineral iron.
The cell sap is less concentrated, and then you think they are losing water to the surrounding. Imagine what is happening around the Lake Choga, what is happening around the plants around Lake Victoria. Eventually, they are going to dry up. Will it be that they are losing water to the surrounding water? Can we hear from another person?
I think too much water in the soil can lead to less oxygen.
Very good. That is where the point is. In the soil, we expect spaces between the soils to be filled up with the air. You remember when you are in agriculture, when you are looking at soil components, one of them was air. And when an area is covered with water, it means that water is going to displace the air in it. And it is this air that contains oxygen, which is taken up by plants, plant roots, which will be used for the process of tissue respiration. So it means there will be no respiration taking place in the roots. And when no respiration takes place in the roots, it's like you, when you have been strangled and no air is taken into your lungs, then no more respiration will take place in your body. So it means there will be no uptake of the mineral salts by the plants because there's no active transport taking place. So eventually, the roots are going to die. And when the roots die, they'll not be absorbing water. and eventually the entire plant is going to die the way it is here. So it goes back to the point of air, especially oxygen, which in the soil will be replaced by water. And when there's no air or oxygen in the soil, then it means there will be no tissue respiration occurring in the roots. When there's no tissue respiration taking place in the roots, then it means the roots are not good. The roots are not going to absorb nutrients, mineral salts, into them. And when there's no uptake of mineral salts, then it means there's no going to be no osmosis taking place, and the plant roots are not going to absorb water. So they end up dying. And when they die, the roots die up, then the entire plant is going to die. So that is what happens here. And that will be where I think we're going to stop, unless you have a burning question.